In this video, we aim to study on the different types of sample preparations based on the various techniques we have conducted. In order to make extraction of aloe vera compound, we use aloin, one of the active ingredients in aloe vera leaf. We have used 5 different methods of extraction by using two common analysis devices, which is GC and Hplc. Besides of that, we want to deliver the types of analysis that is suitable for which types of extraction method. So, without further explanation, I will introduce on the preferable method that we choose for extraction process of aloin. The first one is extraction of aloins by ethanol, and the second one is extraction of aloin by centrifuge method. Those two samples preparation was extracted from aloe vera gel and was conducted using GC analysis, which is gas chromatography. We have chosen 3 different techniques to prepare sample extract from aloe vera gel, and we have used Hplc analysis to run the experiment. It consists 3 extraction method, which is extraction of aloin by aqueous extract, extraction of aloin by ultrasound, and the last one is extraction of aloin by supercritical fluid. All of these extractions are made in order to produce soothing cream based on the aloe vera gel.
GC, it uses separation principle. The mixture of a component to be separated is converted to vapor and mixed with the gaseous mobile phase. Like dissolved light principle, which refers to the analyte's polarity, and the immobilized liquid stationary phase. Polar or ionic tends to dissolve in a polar solvent and vice versa. Non-polar compound will elute first. Then polar compounds will elute last, especially if the column is polar. Then this is the polarity of organic functional group in decreasing order. The most polar organic compound is water. The most non-polar organic compound is aliphatic hydrocarbons. Both qualitative analysis and quantitative analysis are identified by gas chromatography. In qualitative analysis, the purity of sample can be determined. Then the retention time RT in GC analysis can identify the components in mixtures. Then it provides an excellent means of confirming the presence or absence of a suspected compound in the mixture. If no peak was seen in the graph at the same RT as the standard, the identified compound in a sample is absent. Quantitative analysis is the comparison of either the height or area of an analyte peak with one or more standards. Then GC has enormous growth in part to its speed, simplicity, relative low cost, and wide applicability to separations.
What is Hplc? Hplc is a type of column chromatography that pumps a sample mixture or analyte through a column with chromatographic pachymetria in a solvent, or known as the mobile phase, at high pressure at stationary phase. The separation mechanism of Hplc may be split into 4 distinct modes, which is absorption chromatography, partition chromatography, ion exchange chromatography, and size exclusion chromatography.
By ethanol extract from aloe vera gel using gas chromatography. Preparation of the extract by using ethanol. Aloe vera compound, which is aloin, can be extracted by using ethanol extract that were made according to the Legrand process. Firstly, a total of 50 grams of aloe vera powdered sample was mixed with 250 ml of 96% of ethanol. Then, the mixture was stored in tightly sealed container at room temperature for 2 until 5 days, and shaken it for several times per day. This mixture was filtered through filter paper to remove the crude plant materials. The step was repeated for 3 to 5 times before a clear supernatant extraction solvent had been obtained. The filtrates of aloe vera were evaporated to dryness using a rotary evaporator at 40 degrees Celsius. The final dried sample were weighed and placed at -20 degrees Celsius before used. GC MSD-HP 5960B device was used to start the analysis. For the results, a fresh leaf aloe vera were analyzed for these anthraquinones by the initial gas chromatography mass spectrometry method, and the results are shown in Figure 1, where samples are being analyzed and shown the level of aloins obtained. from Figure 1 shows selected ion chromatogram for alloy A at 1.0 ppm. For the internal standard of alloy, ion ratios were used for the identification of those two analytes, which is alloy A and alloy-amidine, to improve specificity of the analysis. Alloy is the main anthraquinone in the alloy leaf found naturally as a combination of two alloys A and alloy B stereoisomer. In addition to these chemicals, compound present was alloy-amidine and alloy A that have been identified with the biological properties of extracts from aloe vera. As a result, alloy A have more amounts of analyte, which are 1.0 ppm compared to alloy-amidine, which contains 0.1 ppm. From here, we know that the amount of alloy extracted by using gas chromatography analysis has been conducted successfully.
Aloe L is a species of succulent plant. It is thick and have fleshy green in color and contains gel inside. Aloe vera gel, a clear jelly-like texture, is used as medicines and based in cosmetics formulation. Anthraquinone glycosides, alloy-amidine, and borbolone or alloy are found in the leaf lining, latex, resin, or sap. Next is methodology. First, the crystallization of crude alloy. The leaves cut into half, scraped, left for 24 hours to extract crude and incubated it at 40 degrees Celsius. Second is the preparation of acute anthraquinones, AQs. It was centrifuged at 10,000 rpm for 30 minutes duration. Then separated into two layers. The upper layer is supernatant clear solution, and the lower layer is the pellet, contained the crude extract. Sulfuric acid and chloroform were added to the extract, and then flux. The chloroform extract was separated by using separating funnel. Then it was evaporated to obtain yellow-brown colloid, and the colloid then dissolved in a methanol. Third is preparing the alcohol or salt aqueous two-phase system, ATPs. Separation phase forming two clear phases, which is the upper phase, small volume of alcohol with AQs, and the lower phase is large volume of salt-rich solution and impurities. Then alcohol-rich phase diluted with 0.5% of magnesium acetate methanol. The aqueous solution color then turned red, and its concentration were determined by colorimetric method by UV spectrum photometer. And lastly is analyzing the sample using GCMS, which is gas chromatography mass spectrometry. The GC column was a 20 meter times 0.18 millimeter. The run time is 25 minutes. Then it reduced to 10 meter column to shorten the run time to 30 minutes. Then it detected at 150.8 degrees Celsius for 1 minute until 280.8 degrees Celsius, which is 30.8 degrees Celsius per minute. An injector at 250.8 degrees Celsius, transfer line at 280.8 degrees Celsius. This is the table. Extraction of AQs in different alcohol, salt, or ATPs. Next is the graph, which is the black line is the sample and the green line is the standard. The sample shows greater peak than the standard, and two main constituents obtained are A at 9.08 minutes and B at 0.5 plus minus 0.05 minutes. Lastly, the conclusion. In this study, centrifugation by gas chromatography mass spectrometry method was carried out to extract a line from aloe vera as soothing cream. Two main constituents obtained are A at 9.08 minutes and B at 0.5 plus minus 0.05 minutes. The peak indicates the presence of a line in aloe vera.
In these methods, we will extract the alpha tocopherol by using supercritical fluid extraction, as alpha tocopherol is one of the compounds in a line gel. The gel and skin of the aloe vera leaves were isolated from each other and instantly frozen for 36 hours to achieve healthy moisture content. The lyophilized powders of aloe vera gel and skin were processed at negative 18 degrees Celsius. The industrial quality carbon dioxide has been used as a supercritical fluid extraction. The alpha tocopherol was obtained from sigma chemical coal and it was analyzed by using HPLC to obtain a standard spectrum. Figure 1 demonstrates the impact of dynamic time extraction and SiCO2 flow rates on the recovery of alpha tocopherol at set 26 megapascal and 50 degrees Celsius. Overall, considering 10 to 20 minutes of static before dynamic extraction, the recovery of supercritical fluid extraction improved due to the allowed equilibrium between solid and fluid phases. Table 4 reveals that increasing dynamic time has a very significant positive linear effect and a significant negative scar effect on R. The positive effect of increasing dynamic time on R is attributed to the higher intake of fresh SiCO2 for alpha tocopherol extractions. Hence, higher recovery can be achieved until there is an efficient driving force of mass transfer between the solid and the fluid phases.
The next title is the extraction of alloy by aqueous extract from aloe vera gel extruding cream using HPLC. The methodology of this extraction. Let's begin with the preparation of aloe vera gel. Firstly, we need to remove the bottom of the aloe vera leaf. Then, the rings or the tiny edges were removed for both sides. The tip of the aloe vera leaf need to remove too. And it will look like this. Use the knife to remove the bottom of the leaf of the gel and then scrub the gel. The top will be dissolved with methanol and lastly the sample will be directly injected into the HPLC system. Next, for procedure in the HPLC system, after the solid was injected into the column, the separation was occurred as the analyte and the mobile phase are pumped through the column. The column will elute each component as a peak on the recorder and the peak will be displayed on the chromatogram as detector response. Let's move to the result for this extraction. Methanol was used in this experiment because a mixture of methanol and water allows the separation of polar components from non-polar components as methanol have a high toxicity. Methanol on mobile phase as a polar compound in a reverse phase chromatography. Increasing the polarity of the mobile phase can increase the elution time. The main reason why HPLC instrument was used because it is useful for identifying, quantifying, purifying the individual components of its mixture. The figure shows the concentration of alloy in three types of solvents respectively after 60 minutes of extraction at 30 degrees Celsius. The maximum extraction using methanol was achieved in other solvents. The effect of the temperature with the extraction of alloy was investigated. It shows that when the temperature increased, the extraction of alloy may increase too. This figure shows a typical chromatogram with solvent peaks recorded by a UV detector. First, collect the leaf from local nursery after rings are removed from the leaves. Next, wash it with clean water for 5 minutes and then rinse with sterile distilled water. After that, the inside gel was scalped, cut into pieces using a sterile scalpel and collect them in sterile container. Then, the sun-dried gel was collected. A long process of drying and dried gel particles were detected at. After that, suspend 2 grams of kaolin in sterile methanol. The solvents used for extraction and HPLC analysis were RHS, AR, and HPLC. Ultrasonic probe was used for sonication. Make sure to keep the temperature constant inside the vessel. Lastly, the
The specimens were taken at different intervals for the whole run and estimated aligned using Hplc. So, this is the result, with an effect of dry particle size and effect of dry gel loading. Now we go to the discussion. For initial 20 minutes, there was rapid increase in extraction in all dry gel suspension loadings. The solid experienced large driving force to get dissolved in the solvent, but at later stage, the concentration gradient decreased and affecting the rate of extraction. The extract equilibrium concentration of alloy, when compared with the maximum amount of alloy present in the dry gel, had shown increasing and at higher loading. At higher loading, the concentration gradient at the solid-liquid interface increased with time as the amount of solvent remains not changed. We can see that from the figure of the effect of dry gel loading show that 7.5% dry gel loading was selected for further experiment as maximum extraction of alloy was achieved. Now we go to the effect of the pressure. This figure shows that the extraction of alloy was increased when the temperature increased, and also can easily be attributed to higher solubility of alloy. And then the kinetic energy as well as diffusivity of solvent increase with temperature, and the solvent penetrate better inside the cellular matrix, leading to the faster release of the alloy molecules. Within 30 minutes, around 19% extraction of alloy was observed at 400 degrees and 92% at 500 degrees Celsius.
As the conclusion, alloy from aloe vera could be extracted using a variety of methods of extraction, such as by centrifuge, aqueous extraction, ethanol, ultrasonic, and supercritical fluid. The results of bits and components present in alloy could be obtained by using two types of analysis, Hplc and GC. Gas chromatography and high-performance liquid chromatography are both separation techniques which have gained immense popularity in both academic and industrial laboratories. When combined with mass spectrometry MS detection, GC-MS can be used to separate complex mixtures, quantify analytes, identify unknown peaks, and determine trace levels of contamination, while Hplc instead requires a small sample size. The test can be modified depending on the level of quantification. We also could obtain an accurate low volume of the sample injected, as it has a good pumping system and higher sensitivity of different types of detectors.